disappointed with how you did, but then you probably already know, like, you might already have a good idea, like, where you might have gone wrong. But at least this will keep you knowing what you need to do when we're in lab today. So all that stuff that we talked about last time with the fungi, you know, hopefully you reviewed that in your lab book. So this lab should go very smoothly for you. But then you know that all that information about what type of pipe you might see, asexual spore and sexual spore, and that will be examples that we'll talk about when we get into lab. You know that you're going to have to memorize that. And any of the other characteristics that I might have just not mentioned at this time, like especially disease or something like that. And just like basically you always know like what's going to be on the quiz. We'll have a quiz. I think we'll have it. We'll have it a week from today. So that means the next quiz will have the culture media preparation, the pure culture technique, you know, the beginning part of stock cultures, the fungi that we're going to do tonight, plus population counts, which we're doing next time, and it's just really critical that you read the beginning part of that lab before you come next time. The lab, and the reason again why I'm saying this in here is because I always feel like I have everybody's undivided attention in here, and when we're in lab, like this little group might be talking, and that noise is going on or something. So it's really like the lab we're going to do next time is extremely important for the whole rest of the class. And it's one of those things where it's some math involved, and it's really how to make dilutions and come up with counts, and it is something that if you're a math person, you'll think it's the easiest thing that we're going to do all semester, but everybody else will have a tendency to get a little bit confused on that, so it takes us a while to explain how to do that. The lab book explains it really differently than I do. And I think, and not just me, but the other two instructors kind of feel like the lab book makes it seem even worse than it is, because they give you all these abbreviations for which things, and multiply this and divide by that, and multiply by this other and divide by this, and it starts seeming like there's no logic to it, but there is logic to it, and you know, hopefully I'll be able to explain it in a way to make it much easier than that. But the beginning part where they talk about why we would count and what other methods there are, and understanding what advantages and disadvantages are of the different methods, that's what you want to make sure that you read, so that you're coming to class already having an understanding of the big picture, and then I'll explain to you how to actually do the dilutions. And it will be something that we will do, you know, for several, like the very next lab also does dilutions. We'll be doing dilutions later on in the semester, and it's just something that is extremely important in microbiology, because when you try to count numbers of organisms, how can you really do that? You know, that's really a challenge because you can't see them. So what are some of the methods that we could use? And this is really going to be one of the most common methods to use. So it's just something that you have to take that lab really, really seriously. So people shouldn't miss that lab. If anybody, you know, had to miss a lab, that would not be a good lab to miss. So don't read the whole entire stuff though, because unless again you had chemistry and you already like kind of get how they do dilutions, you might read it and it might make a lot of sense to you, but everybody else just read that initial introduction to all of that. Yes.
Isn't there a quiz next Tuesday?
Yeah. Perfect. Okay, so as far then as lecture goes, did anybody have any questions about the malaria life cycle that we went through last time? And hopefully you guys are really this close to having it totally memorized. And you know your chapter 1 stuff. How many people have been on that website to look at some of the exercises on there? Okay. 3 or 4 people, because when I go on it, it kind of indicates there are like 3 people have been going on it. So if you go on it, it will encourage you to start to know that material from chapter 1, you know, and the stuff from chapter 17, because I've picked out all the questions. So even though there are some questions on there that are things that I think that you should know, but I wouldn't necessarily ask you to memorize it. So there'll be some questions that you'll come through and you'll be like, oh, I don't really know what that is, and you have to look it up in your book, and now you know something, but it's not necessarily going to be on the test. There are a handful of things like that, but the vast majority of it is actually stuff that you should know. So I think you'll find it helpful. And I did confirm with the other instructor too that they did say that the students will be able to access it for free for the semester. So I just have to, she hadn't heard from that rep either to like find out what kind of code we need to give you guys or whatever. But remember, when you sign up, you get it for 3 weeks for free anyway. So by the time that the 3 weeks is up, I'll have that for you. So keep up with it, because the first exam, if you're not like keeping up with your studies, I promise that you won't be able to learn it all in the weekend before the exam. And it's really like, to me, I'm thinking it's likely that it will be 2 weeks from today. That's my best guess, because I try whenever possible to give the exam on a Tuesday, because it's hard to give you a bunch of material on Tuesday and then test you on Thursday. So whenever possible, I try to do that. So that would be March 5th. And even if it meant like I gave you a little extra material or something like that, we were ahead. But if not, then at least you know right now is like when you need to get all that studying done. So anything before I keep going on? Okay, well, we stopped last time, other than talking about the malaria life cycle, just ending up on the reproduction. The motility that the protists have, so like basically right now we're kind of back to still just looking at protists as a whole, like what types of characteristics they have and such. We talked quite a bit about the organisms that have cilia and flagella or amoeboid type movement. There's not really much more that I really need to say here. We talked even about this idea that we still refer to them as ciliates or flagellates or amoeba type organisms, just because those are kind of significant characteristics. There were other characteristics that went along with organisms that were ciliates and flagellates and the ones that had no locomotion as well, and that's why it really is more likely that we'll continue to kind of refer to them as that, even though that's not exactly what their official classification is. If you look at the structure, this is the same kind of structure that you see in other eukaryotic cells. So when you had your cell and molecular biology class, you talked about cells. So the eukaryotic cells we don't really go much into because you already had your introduction to that. One thing that we did mention in lab is the idea that protists can form these resistant structures called cysts. And that was mentioned just because if you went over to look at the slides, you would see that a slide said that it was a cyst, or maybe it said it was a trophozoite. The trophozoites, is that one? But the trophozoite, I know it had to be on one of the earlier slides. Do you remember that word? At least reading it in the lab? So T-R-O-P-H, tropho, then zoite, Z-O-I-T. That's the sort of active feeding stage. And structurally, they do look different. The cyst, and it says some protozoa, those are, again, like the more animal-like protists, that the heterotrophic ones. Sometimes you might read where it says that all of them form cysts, but all of them haven't even been identified yet at this point. But the important part of the cyst is the fact that that is really critical when it comes to these disease-causing protists, because it makes them so much easier to be transferred from a person to another person. Most of the protists are going to be in somewhat of a moist environment, but then what happens when it dries up? Well, they're gonna be in a cyst, and they're gonna be able to survive like that. So cysts can also survive disinfection. So we get Giardia cysts in our water supply. You saw the video and they talked about the Cryptosporidium. Those organisms in their cyst form can survive the chlorination process. They mostly need to be filtered out of the water. So it's something that when you think about, unfortunately, again, the idea of the fecal oral route and that a lot of these organisms are associated with diarrheal type diseases. Those oral infection species, if that happens to get on somebody's hands or something and gets on a doorknob or gets on a surface or something, those cysts then can survive for long, long periods of time. Not just like minutes or hours, but days like that. So it just really does increase the likelihood that you can pick up these types of organisms if somebody else is sick with one of them. And of course, Giardia being the one that's really the most common here. Also during the cyst form is when you get things like the nuclear reorganization, like we mentioned when we talk about schizophreny and plasmodium.
It's just the Giardia cyst that needs to be filtered out of water, or is it all?
It's all. It's just that Giardia and Cryptosporidium would be the two most common ones that would be likely to be in our water supply. As we go through and learn about these different types of organisms, this information is going to come up again later. When we talk about water and water quality, the microbiological quality of water and water treatment, I'm going to be referring to these organisms over and over. So as you're learning these, don't forget about them. Even though the lecture stuff is not cumulative, it's like it does kind of build on it. So when you learn the different, hopefully, the whole semester will go by, you'll never forget things like Giardia, Entamoeba histolytica, Cryptosporidium, some of those that are really common. And especially if you guys are going into the medical field, these are things that you'll come across as well. So when you look at dysfunction, survival, disease transmission, nuclear reorganization, this would be something that, like I've in the past, asked a short answer about, what is the significance of a protus cyst? Not just like what is it, but what's so significant about it? Well, that's what we're just talking about now. So the idea that that is going to make it much harder to control as far as disease, because they are difficult to kill because they are very resistant. Also that other functions happen during that time as far as that reorganization. So it's called encystment when they form a cyst. It's called excystment when it comes out of that cyst form into a trophozoite. So when we go to talk about the life cycle of Giardia and Entamoeba histolytica, that's part of the life cycles. It encysts, it excysts, and such, so you understand what that means. I think you have this table in your notes. But if you look at these, this is just like some examples of medically important protozoan infections. But if you look at most of these, we've already been talking about Plasmodium. We've talked about Trypanosoma, and we'll talk about these again. We've talked about Cryptosporidium, but it'll come up again. We've talked about Entamoeba histolytica, Giardia. We'll be talking about Toxoplasma, Acanthamoeba, Naegleria, Trichomonas, and Leishmania. So the majority of these we will be talking about in here. So this kind of gives you like a little bit of an overview of what are some of these examples. As we start to go through the examples, and this I don't think that you have this one. This one might be from the previous book, but this is just that chart again that shows. Do you guys have this one in your notes? I mean, it's so small that I know you can't really read it, but it's just in here to list those supergroups so you understand about the classification. But as we go through and talk about the different examples, we'll talk about them kind of in this order. And this is where we're starting with supergroup Excavata. I'm going to want you to know what examples go with supergroup Excavata. I mentioned before that I don't expect you to know those first ranks, but I left them on that paper because That's the order in which they're presented in the book. Also, I have taken chapter 39, which is the diseases associated with the protists and fungi. And I've taken those slides and I've incorporated them into this chapter. So if you tried to go through and look at all these slides before coming to class, because I know that you're getting prepared and looking at things ahead of time, then you'll notice that it seems like things a little bit are out of order, and that's the reason why. Because it's like this first slide shows the list that comes from chapter 23. And then as we go to talk about Giardia, I've taken the stuff from chapter 39 and put in there. So like, that's why it looks like it's kind of out of order like that. I just thought that's easier than like discussing chapter 39 separately. When you look at some of that information, and be sure and like really like listen to what I'm saying about this. I am not expecting you to know like all the treatments of these diseases and all the information about like the mechanisms of how they cause damage to the body and so on. I just want you to have access to the information about those diseases when we're talking about them, because sometimes we get in discussion and people will want additional information, not because they're trying to memorize it for the test, but just because if they understand a little bit more about it because of interest or just because it helps them remember this for the exam. So again, I'm going to be clear cut about telling you what you have to know, but I do have that extra information in there just because a lot of people really are interested in some of these organisms that cause disease. So that's why they're out of order. That's why there's, you know, a lot of information in there that is sometimes stuff that looks repetitive too, because they might cover a little bit of it in chapter 23 and then cover it more in detail. So you do not have to know how to treat things. You do not have to know all the symptoms. You just need to know like some of the basics, like if there's a life cycle involved and if there is information that's obviously like, you know, really related that has to do with different types of symptoms, that might come up and, you know, we'll mention something about that. So don't like try to memorize all that other stuff. So we'll start with Giardia, and of course Giardia intestinalis or Giardia lamblia. That's how you'll see it written. Intestinalis is the newer name, and that's one of the diarrheal diseases that we talked about in the lab. So here you see they're referring to it lamblia. That's why you're always like kind of just stuck with having to know like both names if it's in the process of having those changes. There's just no way around it. So that's why I have to skip through those other couple of ones to get to Giardia. So you see, even though this is the same textbook, here in our same textbook, same authors, and they're referring to it in both different ways. That's, again, why there's no way that I can just like switch over to just making you understand just the one species name. Here, as I think we've already mentioned, it can be in contaminated water. It does form cysts. It's carried by all different types of animals. So not just, you know, human beings, but all kinds of animals. Most mammals will carry it. Dogs and cats can carry it, especially like when, you know, people have new kittens that have been outdoors or the new puppies are regularly treated for it. You see here also they could be asymptomatic. That turns out to be something that, you know, can be important because they have been able to trace back epidemics of Giardia being associated with small children. I think when we were in lab, I might have mentioned something about the whole idea of daycares and preschools and how they had to switch over to being very cautious and following like rigorous guidelines on how to change diapers and dispose of, you know, diaper-related materials, because Giardia was something that was being spread frequently by children at daycares or preschools. So they would pass it on to each other, wouldn't necessarily have any kind of significant symptoms, then they take it home, pass it around at home, and as the epidemiologists come and try to investigate what's going on, everything leads back to one place, you know, that preschool or that daycare center.
How does it get treated?
They can treat it with things like Flagyl and like basically they're called chemotherapeutic agents. It can you can recover on your own as well. And so it depends on how bad of a case you get. It can be something that can be chronic and difficult to treat also. So you see here too, more serious disease in children. So some children can have a lot of problems. Some children can be asymptomatic. It's going to turn out that there's going to be a lot of infections that really can be asymptomatic, and people can be what's called a carrier of a particular organism, which then they can pass on to other people. And we'll get to a point where we're reading about some of those, and I don't remember where it comes up at first, but that idea of Typhoid Mary. Has anybody ever heard of Typhoid Mary, or even just references to Typhoid Mary? Because she was the person in the 1800s that was a cook for different families, and she kept passing on typhoid fever to families, and even way back in the 1800s they were able to trace it back to her, and she never had any symptoms of typhoid fever. But basically in her particular case, they kept telling her that she couldn't work as a cook anymore, because obviously how would she pass it on? Guess how you get it. Take a wild guess. Fecal oral route. So obviously not good sanitary practices. People were getting sick. She would agree that she wouldn't cook anymore, and she would change her name, and the next thing you know, there's another outbreak of typhoid fever. It went on and on and on to a point, apparently her gallbladder was holding on to the organisms, and there was never any record that she ever had the infection. But she kind of became a historical figure just for the fact that she represented this, and because it got all the way to the point where they offered to remove her gallbladder to see if that would make things better, but she didn't want to do that. So she just kept agreeing not to cook. She finally was arrested, she was in prison, she was let out under the guise that she would never cook again. Guess what she did again? Started cooking, and finally she ended up just having to live out the rest of her life in it wasn't Ellis Island, but it was one of those other islands that kind of around the New York area. And they've never really been able to know exactly how many people died because of her or just got really sick because of her. But that kind of thing of, carrying organisms, that are disease-causing organisms, but without people getting sick, something that does happen a lot. That's why you'll see that kind of thing. Asymptomatic carriers, that's what an asymptomatic carrier is. I mean, there's some idea that, is it really that the person has never been sick with it at all? Or is it just that they never got sick enough to have symptoms that were diagnosed? That's still a little bit controversial.
So that we just try to carry.
But just kind of understanding what that meant when it was listed in there. But I mean, that is something that as the semester goes on, we will bring that up again.
Would you consider a carry to a virus or
No, because carriers can be the sick people too. It's just when we're talking about disease, we've never really had the formal introduction to all the terminology that goes along with it. But you can't really talk about diseases and disease cycles without understanding kind of what is a reservoir. I don't know if you noticed in the video that they kept kind of bringing that up, oh, well, there's, all of these different kinds of animals that are reservoirs, so there's always a constant source of the organism. So, just trying to define some of those things as we go along. Because a reservoir would be, whatever organism is, really a place where those organisms are growing and always there. A reservoir isn't always a person who is sick either, kind of like a carrier. So if you look, here's one of these slides that you can see. This slide is from chapter 39. So it's giving a lot of more information about the disease itself, which I'm not going to ask you about, but that question came up, well, what about treatment? And so here they tell you that antiprotozoal agents, how to prevent it, how it's diagnosed. But no, I'm not going to ask you that kind of information. But what I am going to ask you is this, the life cycle. So if you look here, Giardia and Intestinalis, approximately 30,000 cases yearly, and that might even be an underestimate. Like when people are sick, it's not always diagnosed, especially if you have people that don't really have a lot of symptoms. Everybody doesn't go to the doctor. Here is just a listing of the life cycle. So like I talked about malaria with Giardia or Entamoeba histolytica, you could learn it by just listing it like this, or we could diagram it out. Okay, so however you want to do it, as long as you let me know what, that you understand what the life cycle is. This one is just like such a simple and kind of straightforward life cycle that kind of introduces you to life cycles, before we get to the malaria one. Just for you guys, you got to go to the malaria one first. So this one will seem like it's really easy. I think of writing these out, kind of like I mentioned with the malaria one. I think of like, oh God. We have like a real problem with our whiteboards. Did you use up all that?
No, the blue one and the blue one.
I think of kind of my sick person here on one side, and then I think, how are they gonna get it? Where was the green movement? We can only use quartet markers on these boards because like when they built this, they gave us like the cheapest boards imaginable that, believe it or not, just those little expo things actually like ruin the board. So I think of, okay, the person drank a glass of water that had cysts in it, or I think of things like this. You're like, what is that? That's supposed to be broccoli, okay, because it came from a field. So fresh, uncooked vegetables. Oh, I did that terrible drop. That's a way in which you can pick up anything from the fecal oral route, because people are handling the vegetables. They're out in a field, they use fertilizer, there are animals out there, right? When you go through agricultural areas, we already talked about the outhouse idea, right? That now they have them, but they didn't regularly have them, and I'm pretty sure they don't always have them all the time. And they're not fenced, they don't keep out animals, so animals get in there. We've had all these outbreaks associated with things like spinach, fresh spinach, all kinds of like onions and cilantro and sprouts and things, because runoffs come from other areas. When you have agricultural areas, you also sometimes have like farms with cows, ranches and such, and then they think that when it's raining, water runs from those areas and contaminates the fields like that. So you really have to be careful with like raw vegetables. So this person is going to ingest. And so you can see, like, starting up there, they're going to have exposure to the quad nucleated cyst. So this person ingests a quad nucleated cyst. Okay, so gets through the stomach, gets into the intestines, it's going to exist. Okay, and at this point, it's undergone plasmotomy. So you're looking up there and you can see excisment in small intestine, plasmotomy into the quad nucleated into two binucleated trophozoites. So it's plasmotomy, plasmotomy, exis into two binucleated tropes. Okay, at that point then it undergoes binary fission. and then it's gonna continue to reproduce at that point in binary, using binary fission. And if somebody has a really extensive, you know, they've like really ingested a lot of the cysts and they really have an active infection, then you can get a lot of like attachment of the organisms into the intestines, the intestinal walls, those columnar epithelium, and it can really affect like how people digest their food and how they absorb fats, and that's like some of the stuff that leads to what type of symptoms they have. Eventually, some of those then, this is going to be in the small intestines. Eventually, it's going to get into the large intestines, so it's going to encyst in large intestines. You think of like the large intestines as not really as friendly of an environment as the small intestines. It's drier, and they will be more likely then to encyst, and then the cysts exit in feces. And that's how they get like that. So it's really, this is really simple. So when I look at it, I kind of think of the person, whatever the exposure is. That's just like, in my mind, I can memorize that easier than looking at a list like what I gave you up there. However you want to give it, that's fine. Yes.
The nucleated cyst, is that just the name of the cyst, or it has to be at that 4 nucleus time to be, you know, to
Yeah. So like
Yeah.
during that time, like when it's in that cyst state, it's undergoing that nuclear reorganization, so that now it's going to be the quad nucleated. And if you remember that video, the guy actually talked about how like Giardia was kind of cute to look at because it looked like the little monkey face. That's how he described it. I mean, that's where it's helpful to like see and look at these things under the microscope, because then that helps you remember that, oh, this is plasmotomy, because it's going to be a binucleated trophozoite. You know, so again, like as I'm mentioning these things, you should be jotting those things down as like a reminder to you too. You know, that you have that something to connect to that. Yes.
How many short answers do you usually put on the test?
Well, 50% of the exam is like true, false, multiple choice matching, and then the other 50%, so the other 50 points is like essay, short answers. In this case, like there will be this life cycle and the malaria life cycle, and there will be already Koch's postulates and the essay about classification. So you already kind of know that. You just don't know where are those other ones going to be. No, because if you really just take good notes, and as I'm like throwing out like, oh, in a previous exam, I asked this, you'd be surprised how many times that exact question ends up on this exam too, you know? And people will be surprised and leave it blank, and I'll be like, I even told you guys I asked that question before, you know? I try to, you know, ask questions that are gonna show me that you know the information that you're supposed to know. So like, as I'm going through, like I really like, so clearly will say, you know, something like this will be on the exam, or you should be able to, like I already said, right, about one other one, about the significance of the cyst. So, you know, like you wrote that off to the side, but if you guys don't, then you think like 3 lectures from now, you won't remember that I said that. So I'm just telling you, like, it's not just me, but other people that you talk to that have had this class, or going on Rate My Professor, they'll all say the same thing. If you take good notes, you're gonna have like already a good idea of what kinds of things are gonna be on the test. So I don't like give you like an outline. So really at this point, there's other organisms to talk about in excavata. And hopefully you're not going to really hate this, but I am going to go right over to Entamoeba histolytica, and you'll see why. So Entamoeba histolytica, the life cycle is so similar to Giardia that now that we're talking about it, it's so much easier to have this life cycle at the same time. Okay, so if you look at this, and I think I mentioned this in lab 2, third leading cause of parasitic death, and this is in the world. And it's a significant cause of, and this is slide 34 here, having trouble finding it, but this is a significant cause of infant mortality in developing countries. Entamoeba histolytica is so much more of a serious disease than Giardia. And Giardia can be serious, and one of the things that I mentioned when we were talking in lab that you might have written down is that part of the reason that it is so serious is because it is an invasive organism. So you see here, may migrate to lungs, brain, liver, or skin because simply what this organism will do is burrow out of the intestines and get into other parts of the body. So if you look at the life cycle here, this is the same. It's the fecal oral route. The person is going to ingest the cyst. Let's see if I can just get to it. So it's also a quad nucleated cyst. Or okay. It doesn't undergo plasmotomy, but it undergoes schizogony. So remember what schizogony was a multinucleated cyst to single nucleated cyst. So in here you have a quad nucleated cyst. If it undergoes schizogony, now you're going to have 4 uninucleated trophozoites. So I can take like the same drawing and just like switch it right over to Entamoeba histolytica. And that's why I say for you guys, you can choose one or the other. I'm going to have the blank there. I'm just going to say diagram and describe, or just describe the life cycle of either Giardia or Entamoeba histolytica. You still have to know about the organism, but you just have to commit to memory one of them, which obviously you can see if you know one of them, you actually know the other one anyway. But in here, when you have binary fission occurring in the small intestine, at this point it could be invasive. You'll start to see that every time we talk about some kind of infection that's invasive, it means that bloody diarrhea is going to be a likely symptom. So when I said, oh, you don't have to know about all the symptoms and blah, 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 but maybe if there's something that's significant, that is a significant symptom. Because as we go through and we talk about bacterial infections also, the same thing will happen. If somebody has something like bloody diarrhea and they have like some kind of evidence of having a bacterial infection, we start thinking of Shigella, E. coli 0157H7, the one that we've kind of mentioned now a number of times. It also is invasive. So those are the types of symptoms. So if you are a clinician, if you're working in the health field, somebody having bloody diarrhea, that is a sign of an invasive infection that's already going to start, narrowing down the search of, what somebody has before they actually have to go through and culture things. It turns out that it's very hard to culture a lot of these organisms. If you're looking at something like a protist, then you are going to look at a stool sample and look at it under the microscope and see if you can find those cysts. But that means that you have to get the right amount, you have to get the part that actually has cysts, so somebody probably has to have a more extensive infection. If they have a bacteria and they're being treated with an antibiotic, the organisms might not be there to be seen. So there's all kinds of difficulties in diagnosing some of these infections. So having something that's invasive, that's going to be a sign to help a clinician start to figure out how somebody is going to be treated. Okay, otherwise everything is the same. So again, in that case, it's definitely worth understanding a little bit about that particular symptom.
It grows out of the small intestine or the large intestine?
it is continuing to undergo binary fission in the small intestine. The large intestine you think of as kind of a less hospitable. You know, even though things could still grow there, but it's just gonna be less of a moist environment. Okay, so any questions about that one? So let's see if there's anything else. Here's like something, this is again from our textbook, kind of showing, you know, excisment. They call this part a metacyst when, you know, it has the quad nuclei, the trophozoites. When I look at this, to me, that doesn't really make me feel like, oh, I'm gonna be able to memorize this easier. I personally, even though that might look like a mess to you guys, in my mind, like I just kind of can see it. It makes sense. When I look at something like this one, it just doesn't look that easy to me. It looks like, again, like how that one malaria, that's just me. If you guys liked it like this, you're welcome to write it out like this. Anyway, like I'll be able to follow it. I know it at least well enough like that. Here it shows you kind of a good picture of what a cyst looks like and what the trophozoite looks like. But remember, this is an amoeba, so it doesn't have like quite the same kind of structure as you'd see for Giardia. And if there's anything else about this one. Again, you can see this is like from the chapter 39, so you can see how the format of it is a little bit different. Talking a little bit about make sure you close that door. So see here, exhaustive diarrhea, appendicitis, abscesses of the liver, lungs, and brain, you know, something that, again, if the organism gets out and gets someplace else, you know, can be so much more significant. So we kind of go back to that first slide, sorry, back to 14. We talked about Giardia. We skipped ahead to talk about an organism that was in the supergroup Amoebozoa, and now we're at Trichomonas vaginalis. Yes. No, it's the same. It just has a different name, but like so many other things, they've never like totally quit using the old name. Oh, if Giardia lamblia and Giardia intestinalis are the same. And that's why I have a tendency to just keep like calling it Giardia, you know, because it is, I understand it's confusing. But I also understand that you guys, again, just have to know that that's the way that it is. And you're gonna see this like forever now. Since you guys are going into the medical field, you're gonna see this and be like, what is this organism? And then think, oh wow, I learned this as, you know, when I was in school, they called it, you know, something different. And it's just because of the things that we talked about in classification. There's so many changes, there's new techniques, there's ways of identifying organisms that now they realize that, oh, okay, it should really have this other name because it fits more with this particular group than that particular group. So Trichomonas vaginalis, this was one that you also looked at in that class. And with these two, I'm not going to ask you to write out all the names, but just as a warning, don't not learn them as well, because you think you'll just recognize them. Because like Trichomonas looks a lot like Trichonympha, and we're going to talk about that organism too. So if you just like kind of get on that idea that, oh, I'll just remember that one, I'll just recognize it. So kind of study like as if you had to write it out. But I'm not going to ask you to write it out. So with this organism, this is the one that is associated with an STD, and it's extremely common. Look at these numbers, 7 million cases in the U.S. There's actually like 7 million new cases every year, 180 million worldwide. So this is something very significant. definitely, and I think here you can see like some of the information that's from that chapter 39. Let's see, where was that part? Oh,